to remind us on the embryology of the brainstem and see whether we can learn from murine and viral models so, Sebastian, all yours.
My research has always been directed to understand basic mechanisms of signal transduction, and because firstly I come from insulin and IGF-1 signaling field. I did most of my work in Joslin Diabetes Center studying insulin signaling. Then I switched a little bit to neural development, and I got very interested in signals that make the cells grow and differentiate. And this biological problem has been conducting me through different systems and models where I've been trying to answer the questions I've been generating. Okay, when Jaime proposed me to speak, to talk in this meeting, I didn't know a lot about these brainstem tumors, and I started reading, and I read almost everything I found in the literature, and I found it really exciting because my field was very much restricted to other kinds of tumors, and I never tried to find a correlation between those and definitely another kind of tumors like glial tumors. This one. Okay, first of all, I know that Dr. Cruz already explained a little bit the structure of the central nervous system and of the, but I like to point out certain things because it's different if we talk about the anatomy, if we try to look of how the system works or how the system has been formed. And then we like to divide the central nervous system in different regions that somehow can explain how it developed. Then in this cartoon we can see that we can separate the brain in different areas like telencephalon, diencephalon, mesencephalon, metencephalon, myelencephalon. This is an adult brain. And when we talk about brainstem, we normally talk about certain areas like diencephalon, mesencephalon, metencephalon, myelencephalon, and they are all because they are all located very close, not because they have a common function or so. Some authors don't like to include the diencephalon in the brainstem. Some others do. It's not very important. And there are other nomenclatures, mostly coming from development areas, that call forebrain, midbrain, and hindbrain, that somehow also can explain development processes. If we look closer of what are the structures that we can find in the brainstem, we see that we have diencephalon, we put it or not, and we have mesencephalon, the pons, the cerebellum, and the myelencephalon. All these areas really have not a lot in common. It's an area where we have a lot of nuclei, and especially what we have is a lot of tracks, ascending and descending tracks that mainly go through these areas, and some of them relay in a nuclei or just go through. And It's just because it's the only way to go down. And as Dr. Cruz said, this area contains the output of all the cranial nerves, and in this case, in this tumor we're talking about, most of the signs and symptoms we have is because they press or they disrupt one of these nuclei or one of the outputs. If we have a dorsal view of the brainstem, we can see we also have Brilli areas like colliculus or geniculate for auditory and visual system. We also have descending and ascending sensory and motor tracts that go through this brainstem area. Here we have a ventral view where we can appreciate the huge mass that represents the Pons, because Pons is basically a Brilli area. Okay, this is a little bit to introduce where I want to focus my talk. Brainstem tumors account for 10 to 50% of the child tumors. I'm not including medulloblastoma here because medulloblastoma can account in certain ages up to 25% itself. And if we just look at how they grow, we can talk about endophytic or intrinsic, is the same, and diffuse. Endophytic and focal. Exophytic, and it means it grows towards outside, and the cervicomedullary that can be considered apart. Definitely the one that that we have to pay more attention is the endophytic diffuse, because although there are other tumors growing in this area, like we have the focal brainstem tumors, they used to be low grade, and they used to grow in different areas of the brainstem, mainly in the tectum or in the cervicomedullary area, or even in the Pons. But as I said, these tumors normally are pilocytic astrocytomas, and they have a good prognosis. Unfortunately, most of the tumors that grow in this area, in the brainstem, are diffuse brainstem tumors. And unfortunately, they grow mostly in the Pons. They are the DIPGs. This kind of glioma is really aggressive. I just wanted to mention that medulloblastoma is also originated in this area, but I don't consider, I will talk a little bit later on this tumor, but I don't consider they have the similar origin, cellular origin. So then I wanted to try to talk a little bit, not to understand because nothing is known about that. What's the cell that originates these diffuse interpontine gliomas? And reviewing the literature, it's impossible not to make this to find this similarity. It's, as you already said, the prognosis of this tumor has not improved in many years, and it remains really low, and it resembles a lot glioblastoma multiforme. I'm not talking about histology, I'm just talking about the way it grows and the way it behaves when the tumor is treated with chemotherapy or radiotherapy. Though it's the question that arises is could you find a different cellular type that originates both tumors? And we have arguments in favor and in contrary, and an argument in favor of this thesis is that both tumors are really invasive. They are radio and chemo resistant, and they, of course, grow very fast and have a fatal prognosis. The arguments that go against it is that the diagnosis ages for both are very different, because if we talk about the DIPGs are mainly diagnosed around 6 years old, and glioblastoma multiforme is mostly diagnosed after the 6th decade. It's really different. And the area where it appears, and glioblastoma multiforme appears mainly in the hemispheres where the DIPG appears, as you know, in the Pons exclusively. So I wanted to introduce a concept here that is not new, because if you review the literature, this concept is already proposed more than 100 years ago. Is that what is the cell that generates most of the tumors? Because it is an issue that has been in debate for many years. In brain, we know that we have neural stem cells that during development occupy most of the brain, and then they remain in certain areas of the brain, in the adult brain. This is relatively new, the presence of the neural stem cells. But cancer stem cells, the concept is not new. It has been known for a long time that some tumors present different areas where some areas look like more undifferentiated, others are more differentiated. But in especially because the presence, the discovery of good markers to label stem cells help a lot to understand how the progression of a tumor works or can be explained. And if we have that there is a huge similarity between neural stem cells, for instance, and certain groups of cells that we can find in most of the tumors, not only brain tumors, because they have been found in lung tumors, they have been found I read liver tumors, and they will be found mostly in almost any kind of tumor. The point is that this is not working anymore, but anyway, the tumor contains a certain number of cells that do have markers of pluripotential cells, mostly stem cells, like CD133 or nestin, whereas the rest of the tumor presents other kinds of markers more related to differentiation markers, like GFAP or neurofilaments. So the point is the tumor generated because the neural stem cells are mutated, and then part of the tumor differentiates into another species, or some of these neural stem cells differentiate in a committed precursor, and then a mutation in these ones gives low-grade gliomas. This is not clear yet, but I think more and more data is accumulating to confirm this pathway, that most of the tumors are generated in a cell that is multipotential, has a high mitotic capacity, not activity, because one of the characteristics of these cells is that they can grow very slowly and they are really resistant. The presence of cancer stem cells has been demonstrated in many tumor types, including neuroblastoma, melanoma, and glioblastoma multiforme. And the most interesting characteristic I think is that brain tumors share a lot of similarities with these cancer stem cells. One of the most interesting one is that these cells has a lot of capacity to repair DNA. It means most of the cells, when they are where the DNA suffers an aggression, most of the cells with alkaline chemotherapy or with radiotherapy, most of the cells would make a checkpoint at G2 and they say, okay, this is wrong, I'm gonna die. But these cells just don't do it. They just stop, they take their time to repair the DNA and they can go on. This capability could be one of the explanations why some kinds of tumors are just not, they are just radio resistant and chemo resistant. Okay, what about the location? Location is important. Where is the location of these stem cells compatible with where the tumors are generated? Okay, this is in adult, and this is a rodent or any kind of animal that do have the smelling capacity very developed, where this is a very important way to generate new cells. But that's not the case in humans. Humans, fortunately or unfortunately, I don't know, don't use the smell sense very a lot. But in humans, what we have is certain areas that are important to keep ongoing during all the lifespan. I mean, we have telencephalic areas, or in the temporal lobe, we have the hippocampus, where it's very important to renew the cells. So the presence of neural stem cells has been demonstrated in both areas, in the subventricular area of the hemispheres, in the lateral hemispheres, and in the subgranular area of the hippocampus. Both areas do have neural stem cells capable of proliferating and migrating and giving new neurons that can take over the function that a neuron that was already there degenerated. So that was in adults. But what happens during development? During development, we don't have that areas called neural stem cells areas, because all the tube, this is the neural tube, then it falls and it makes a primary germinative epithelium that gives rise a lot of neurons. It's a very proliferative, very active. Here we can see that this epithelium can give rise to cells that are already committed, and they can migrate out of the epithelium, and they can give different things like the neural crest. We know since a long time ago that most of these movements and proliferations are controlled by signals that come from the dorsal plate and from the ventral areas. And as I told you, we have here the neuroepithelium that is in contact with the ventricle. It would represent the adult neural stem cells that is generating new neurons that move towards a most lateral location, and here they are committed, but they are still conserve a lot of growing potential and migration potential. I have a nice picture that is a gift from Dr. Martí. This is GFP. Okay, these are somites. The somites, it's a little bit, it should be a little bit darker. The somites, half of the neural tube has been transfected with GFP drive it, by a BMP response element. It means all the cells that you can see in green is because they are responding to BMPs. You see, this represents the cells that they are delaminating, and they are forming the cells that will give rise to all the organs formed by the neural crest. It means all these cells are intensively responding to an order given by BMP. So if we look at how the brainstem is formed, it's nothing else than the neuroepithelium. It proliferates, and it generates a certain layers of cells that will be responding to signals coming from up and down, and they will locate in different areas, and then they will connect to other areas, and then finally they will form a so complicated structure like is the brainstem. This is a drawing of the rhombencephalon, and this is the cerebellum primordium, and here is what we call the rhombial lip, and this area will give rise to the pons. And then if we look at what happens here during the brainstem development, we'll see that all the cells generated in the middle part where the epithelium is will have to migrate towards the side, and then will divide again, and will give rise to all the pontine nuclei that finally will connect to all the tracts that come from mainly from the cerebellum, and will have to fit in between all the tracks, the ascending and descending tracks. This is just to illustrate the huge bundle of tracks that connect the cerebellum to the pons. If we cut an adult pons, and we can consider a child is an adult in this aspect because the development finished, we see that we have a lot of these dots represent cells, and to be the nuclei, the cells, the neurons, and all these tracks are commissural tracks. They're going across, and all the other are descending tracks or ascending tracks. What I mean is the cells that differentiate into the pons get compartmentalized between a jungle of tracks, ascending tracks, descending tracks, and different structures. And I want to put the question I started with is, we have tumors like glioblastomas, and they have neural stem cells that can be originated in the subgranular subgranular zone or subventricular zone, and they grow in the hemispheres, basically in the hemispheres. We have other germinative areas in the development, and some of them are very active in the area that will give rise to the pons. Is there a connection between them? Can we study it deeper? Can we make a model where to study if the question is a cell mutation gives rise to the tumor, or maybe just the cell is not in the right environment and it doesn't receive the right signals to differentiate or to stop, or maybe most of the mutations we find in a already formed tumor would not progress if the environment would be the correct. And now I'm going to explain a little bit what has been my work trying to study what makes the cells. That it's not the same system, it's a cerebellum. The model we have been using is how these cells is the dorsal part of the brainstem in this case, and most of the cells come from the same areas. The difference is we have in cerebellum we have a germinative ventricular zone that is really the first germinative epithelium that gives rise to a certain population of cells that will generate the different cells that we can find in the adult cerebellum. Some of these cells move towards its final location, and then they just differentiate. But other populations undergo a migration, and then they can do a second wave of proliferation. And then we thought that was a good model to study what signals would control this migration and posterior proliferation process. I don't know if the scheme is not clear for you, but this is, as you see, the anterior part. This is a cerebellum primordium, and the anterior part is more differentiated. And as we go back to the tail, to the posterior, the cells are less differentiated, and finally what we find is the cells located in the secondary epithelium and ready to proliferate, but already they didn't do anything. And as the arrows mentioned, all the cells generated in this ventricular zone will move in this way to occupy the whole cerebellum cortex. Several groups found out like almost 10 years ago that the signals that control the proliferation of this secondary epithelium, this would be where the granular cells locate, and then they grow again, a second wave of growth that gives rise to a huge number of granular cells that we found in the adult cerebellum. So this secondary growth was mainly controlled by a morphogen called sonic hedgehog. This factor is produced by Purkinje. Purkinje are these huge cells here, and then it goes towards the granular layer and makes them proliferate in a very active way. So our question has always been, okay, these cells proliferate here. At certain point, they stop proliferating. But if you look at sonic hedgehog production, it doesn't stop production. I mean, sonic is produced even in the adult, but at certain point, these cells know that they have to stop, they have to migrate, and when they reach the final location, they have to differentiate. They don't have to grow anymore. And this is I like to put this drawing because it's Cajal's drawing, and he could predict all the cerebellum function just looking at how the circuit was organized, and he describes a lot of characteristics of these cells even better than they are described now. Okay, the first question we wanted to answer is that if it could be the environment that makes the cells to differentiate, because these cells actually, if you take them out and then you put them in culture and you put artificial sonic hedgehog, they don't stop. They grow and grow and grow. I mean, they are not transformed. If you keep them like 20 passes, they finally they will die because they don't have unlimited potential. But you can keep them a long time in culture. So we thought maybe it's the growth pushed them down, and then they reach an area where the environment is not as good as it should be for growing, and it maybe is more a differentiative environment. We look at the different extracellular matrix proteins we could find there, and we found that laminin was there, vitronectin was also there. This is the receptor for laminin, and this is the main receptor for vitronectin. And they had interesting patterns of expression. And so we move on, and we looked at if they had any effect on proliferation. And yes, they really modulated proliferation. In parentheses, if we coat a plate with laminin, the sonic hedgehog is like three times more powerful. The proliferation is more intense. The cells grow faster, and the culture can be more compact. It's really a growth proliferation factor. And contrary, vitronectin mostly induces differentiation. This was done in Madrid. It's already, and we could at that time we could connect that signal with CREB phosphorylation. We could never demonstrate that vitronectin really activates CREB phosphorylation or any other signaling molecule, but the presence of vitronectin kept the cells in a, let's say, high CREB phosphorylation status that modulate them to differentiate. It's not that vitronectin induced a rapid, quick differentiation, but it really helped. And in many aspects, this tendency to differentiate can be very important to stop a cell to grow. This is just a confirmation that with a dominant negative form of CREB, the anti-proliferative, let's say anti-proliferative function of CREB was reversed, and even we had a stimulation of proliferation. This is I think a nice picture to illustrate how thick is the mesh of fibers a cell has to go through to reach up. We'll find the proliferating area, the granular area. Here are the Purkinje cells expressing sonic hedgehog, and all these fibers are absolutely coated with vitronectin. Any cell that has to reach the final location will have to go through all this vitronectin-rich environment that will be modulating the proliferation. Of course, vitronectin is not the only signal that can be modulating proliferation, and in that sense, one of the signals that is produced in the roof plate and in the dorsal areas that is known to modulate proliferation in the neural tube is BMPs. So it was logical to try if BMP could be regulating the proliferation induced by sonic hedgehog on these cells. And yes, both BMP2 and BMP4 that mostly reach the same receptors. I mean, they are two molecules. They have different locus and regulations, but they have mostly the same effect. Both reverse the proliferation induced by sonic hedgehog in these cells. And another interesting thing that at that time we didn't even mention in the paper, we put the picture, but we tried to avoid to talk about that because we didn't understand very well, is that, yes, BMP2 stops proliferation. We didn't see many cells proliferating, but we saw certain clusters of cells that were not astrocytes, typical astrocytes of the culture, but they were GFP, GFAP positive. I mean, glial fibrillary acidic protein. It's a glial marker. Now we know that these cells are already are also CD100 positive. It means these are the remaining uncommitted cells that these cells, the precursors that are already well committed, they have no chance. BMP make them to stop proliferating and they cannot do anything else. But these cells that still retained fate potential, in this case BMP, differentiate them into glial cells, not into neurons. And I think that's very important, and this I will mention it later. 2 minutes, okay. So, BMPs were located in the right place, fortunately, and when we look at the receptors, because BMPs use a transduction, system that uses SMATs, transcription factors, to transduce the signal, and SMATs were phosphorylated as expected in the right place. This is the work done by Ruben mainly. So finally, we have been investigating what is the way that BMP2 uses to control proliferation. And you'll have to believe me, because I removed most of the raw data, if I tell you that we have demonstrated that BMPs, through the activation of SMAs, of course, induces the production of two factors that both, and through a very different mechanism, control the activity of the oncogene and MYC. One, that is TF1, is a Klippel-like factor. What he does is basically he stops MYC transcription, and the other one, and I think is more interesting, is MASH1 is a proneural gene, and we know that it has been always the theory that proneural genes could be acting as anti-tumorigenic genes, controls the occupancy of E boxes in cyclin D2 promoter. That cyclin D2 is one of the markers always uses to analyze proliferation in this. This is only the data. This is a model that would explain, and that's it.